I'd like to thank the conference organizers for giving me this privilege of sharing with you our recent advances in DNA nanotechnology. I haven't had any connection with Govind Khurana, but nevertheless, it's a great honor to be able to participate in this tribute to him. As Ned mentioned, we owe him a debt of gratitude for his pioneering work in figuring out chemical synthesis of DNA. Although, as he also alluded to, we've managed to not take advantage of his pioneering work in figuring out the genetic code. But maybe someday we'll include that as well. So Ned Seaman has taught us over the years how a very simple set of rules, including multiple crossover junctions that he glossed over in this presentation, allow us to treat DNA as a programmable, self-assembling clay that we can mold into almost any desired shape. So for example, this is well, I should mention that my laboratory is interested in asking two questions. First, how can we push the state of the art in assembling DNA devices of ever-increasing complexity? And secondly, how can we employ these devices towards useful ends? And to exemplify this is a transmission electron micrograph, if we could have the lights dimmed a bit, of actin filaments in vitro alongside artificial DNA nanostructures constructed in our laboratory. These actin filaments are about 10 nanometers across. Here's a blow-up of the DNA nanostructure. It's made entirely out of DNA. And our goal here is to capture actin tetramers into these mandible-shaped cavities, and then to polymerize these protein DNA chimeras into an artificial actin filament, and to use it to study the biophysical mechanism of actin-based motors. And my goal is to use examples like this to persuade you that we've made great progress in extending the DNA origami method that I'll explain from the flat structures, flat two-dimensional structures demonstrated by Paul Rodeman a few years ago, now to solid three-dimensional objects of a size and complexity comparable to that found in small viruses. This DNA origami method involves starting with a long single strand of DNA of known sequence as a scaffold. Usually it's the 7,000 nucleotide genome of the M13 bacteriophage, and that's indicated here in gray. And then what we do is we chemically synthesize hundreds of oligonucleotides that are programmed by sequence complementarity to act like clamps that staple together the scaffold into an anti-parallel array of helices. And you get this to work by mixing the strands together, heating it up to a high temperature, and then cooling it down slowly to room temperature over the course of about an hour. And you end up with this parallel array of double helices. Adjacent double helices are held together by these holiday junction crossovers that Ned just described. So Paul Rodeman used this method, and one of his famous demonstrations was to build this disk with 3 holes. And this is an atomic force micrograph. It's about 100 nanometers across. This example up here represents just the area, part of the upper lip of this object. So this is quite large by macromolecular standards. It's like if you take the mass of 2 ribosomes, treating it like molecular silly putty, and then smushing it out to create any desired cookie cutter shape. Here down below you can see the routing of the scaffold through the structure. So when I saw this, I was really amazed. And we wanted to make a contribution by extending this method to 3 dimensions. So how do we do that? Well, the basic idea is that we want to fold the structure. And it turns out that it's easiest for us to fold the structure along the interfaces between the helices. And I'm not going to explain. Hopefully you can take it as an article of faith. If we simply shift around the position of these holiday junction crossovers, we can create curvature along that axis. So I'm going to give you a demonstration here. Imagine that every cylinder is 1 double helix. It's just like this, but rotated by 90 degrees. This is the routing of the scaffold through the structure. But for the purposes of this explanation, I'm just going to leave invisible the scaffold and staple strands. So what we do is we shift around the position of those crossovers so that this sheet of DNA no longer prefers to be flat, but instead prefers to curl up into this S-shaped structure. Furthermore, with, in particular with a honeycomb packing, so we have these cavities. And this looks quite nice just with this simple simulation, but before trying the experiment, one does have concerns that it could be more difficult than achieving this flat, more two-dimensional structure. So in this case, you could imagine it's very complicated. Here we also have staple strands that traverse all 3 layers. So the crossing pattern of those staple strands is much more complicated for this 3D object than for the 2D object. We're packing a lot more negative charge into a smaller space. And so before doing the experiment, one doesn't really know whether it'll work. So we went ahead and tried it out. And perhaps not surprisingly, it didn't work. And that was very depressing. But we were persistent. And eventually, Hendrik Dietz in my lab had the key insight, which is not that these structures don't fold, but rather that they fold very slowly. You have to be patient. So these guys, if you heat them up and cool them down over the course of an hour, that's enough. For these guys, we have to heat them up and cool them down over the course of about a week. And at the end of that process, we get a yield of maybe 5 to 50%, which is a heck of a lot better than 0%. So we were quite happy to find a way to get a reasonable yield of these structures. So Sean Douglas in the lab, who made this very nice movie, decided to fold this structure, but with 10 layers instead of just 3. So if you look closely, this is actually 10 layers of that honeycomb lattice passing back and forth. Sean was a graduate student when he did this work, co-advised by George Church and myself. And now he's moved on to a position as a fellow at our Wyss Institute at Harvard. And so what you do is you order the staple strands, mix everything together, heat it up, cool it down, look at it by transmission electron microscopy. This is with urinal formate negative stain. And lo and behold, you see particles that very closely resemble the three-dimensional structure that we have designed. So in some cases, the particles stand up on the grid surface. You can see this hexagonal pattern of holes. Most of the particles actually sit down on the grid, and we can see characteristic patterns of negative and positive that correspond to our expectations. Sometimes they even stand up in a Leaning Tower of Pisa type orientation. And so now what I'm going to do is to show you a series of different shapes to persuade you of the generality of this method for fabricating three-dimensional structures. This is the structure I just showed you. We call it the monolith. It has the predicted dimensions of about 12 nanometers by 36 nanometers by 36 nanometers. This scale bar here is 20 nanometers. Here's a structure built by Francisco Graf in the lab. We call it a genie bottle. Again, the scale bar is 20 nanometers. And we call it a genie bottle because in one version, not depicted here, we actually left a bunch of unfolded scaffold coming out of one of the ends. So it looked like it was reminiscent of wisps of smoke coming out of a bottle. This is another structure designed by Sean Douglas. We call it a square nut. It has a cavity that's about 6 nanometers across. Again, scale bar is 20 nanometers. This structure on the right, built by Tim Liedl in the lab, we call the railed bridge. Again, every cylinder is a double helix. Scale bar is 20 nanometers. And I'd like to point out here that along the helical axis, we have a different arrangement of double helices. So hopefully you can take this example and imagine how one could generalize to almost any three-dimensional structure using this method. Here's a more complicated structure built by Björn Hogberg in the lab. We call it the slotted cross. You have an O domain and an H domain. And here he's programming the middle of that H domain to thread through the O domain. And remarkably, this is all folding with just a single scaffold. And he doesn't direct the kinetic folding pathway at all. He just designs the final equilibrium structure, closes his eyes, and then when he opens them up a week later, he sees these objects here, which is quite remarkable. Here's another structure. I'm not going to dwell on it very long, also built by Hendrik Dietz. We call it the stacked cross. We have these two domains folding from a single scaffold. We even have a tiny little cavity on the inside here. This is actually the same, related to that initial structure that I showed you, but this domain here with the small cavity is rotated by 90 degrees with respect to the original one. I'd like to now go back in a little bit more detail over this slotted cross design, just as an example. So here's a stylized representation of just the scaffold. It's routing through the structure. We can see that the H domain and the O domain are only connected at 2 points. So we have 2 scaffold strands crossing over here, and then we have 2 scaffold strands crossing over here. And this is what we call a strand diagram. It's what we think of as our blueprint, where we smush out all of the helices onto a two-dimensional surface. For those of you who are close, you can probably even see there's some sequence here. This describes all the information about the object. And it represents just the upper left-hand corner of this H domain. If we zoom out, we get a sense of the scope of the object. And when I look at this, I have 2 immediate impressions. So one is I'm reminded of those old cereal boxes where you could cut something out from the back and then fold it into a 3-dimensional structure. I mean, these things, even though there's a lot of detail here, it really does look like some kind of blueprint. And the second thing that really jumps out at me is that Although the principle is simple, it's actually a pretty complicated object. You have to keep track of 7,000 base pairs in 3 dimensions, which is quite tedious. And the way that we designed all of these structures was we wrote custom computer programs to help us, so that certainly was a step up from just drawing things in sand, which we could have done, but not as good as what we really wanted. So even with our ad hoc computer programming approach, we have this problem where there's quite steep leaning curve. So it's not surprising if people would be intimidated by trying to reproduce these kinds of designs. And even for people who are experts in our lab, doing it in this way is very tedious and error prone. So we had to pay a lot of attention to not making mistakes. And again, even for the experts, it would take probably weeks for us to design one of these structures. And for this reason, Sean Douglason Lab undertook an effort to write computer software with a graphical user interface in order to automate a lot of these tedious processes. And on the advice of George Church, actually, we decided to call it CAD Nano, which I think is a cute name. This is open source software. You can download it for free at this website. There's also a link on our lab website. Sean was the lead developer. He had a lot of help from a Yale undergraduate, Adam Barblestone, who's now a first-year Harvard biophysics student, and some undergraduates from MIT as well. And I'm going to go through a demonstration just to show you how simple it is to design these 3D structures when you have software to help you. So in this example, Sean's going to show us how to build a 6-helix bundled DNA nanotube. This architecture was originally invented in Ned Seaman's lab, and we adapted it to DNA origami. And we're going to just make a 14 nanometer section of this object. So we have 3 panels in the software. We're looking down the axis of the helices. And the first thing is he says, OK, I want 6 helices on our honeycomb lattice. We can see our strand diagram here. This is a 3D representation. And now what he's doing is he's extending these scaffold strands. And we can see in the corresponding 3D view that the structure is growing in 3 dimensions. But really what we want is the scaffold to fit on just one strand. So now what the software does is it forces you to insert your crossovers between the strands only where they're allowed according to what makes sense in 3 dimensions. So this is really nice. As a user, you don't have to pay attention to these tedious geometric details. And maybe more importantly, it's really hard for you to screw it up because the software is constraining you. So this is just the scaffold strand. Remember, with DNA origami, you also have staple strands. But once you've described the path of the scaffold through the structure, it turns out the path of the staple strands is determined based on the simple geometric rules of what DNA looks like. And for that reason, we can add all the staple strands, no matter how complicated your scaffold diagram is, by simply pressing this one button here, auto staple. And like that, in just a second, you have all the staple strands. The next step is to insert the actual scaffold sequence. So you have to insert a break in the scaffold strand. And now what you can do is you can cut and paste your favorite sequence just as the ASCII sequence or one of these preloaded sequences into the buffer. And then voila, you have sequences of oligonucleotides that you can now email to your favorite oligonucleotide vendor, wait a few days for them to come in 96-well plates, and then get your folding started. The program also outputs the strand diagram in a vector diagram, so you can look at it in your favorite vector program, for example, Adobe Illustrator. I'd like to mention that we're quite happy that we actually have one application that really works. It turns out that these DNA nanotubes are useful for solving the structure of membrane proteins using solution NMR approaches. And I don't have time to explain to you how this works. It's based on the idea that the nanotubes will naturally form dilute liquid crystals when you concentrate them, and that's useful for aligning the membrane proteins. But if anybody's interested, I'd be happy to talk about this more offline. So to test this software, we wanted to enlist the help of a summer intern, Katrina Galkina, who didn't have any programming experience and didn't have any experience with DNA. And we wanted to get her to build a structure like this as a symbol of our project to study how the shape of particles affects the rate of entry into cells. So she used the software in just one afternoon. She was able to design this structure here. We ordered the strands. They came in a few days. Heat everything up together, cool it down, after a week, look at it using transmission electron microscopy, and this is what you see. So they look like little cave paintings. I'd like to point out a couple structural features here. The head is attached to the body with a flexible link, a hinge, and the tail is connected to the body by just 2 double helices. Here's a close-up on a couple of these objects. And we can see that the points of weak linkage correspond to some conformational heterogeneity. So you can see this particular Trojan plasmid is happier to see us than the other one is. If one wants to generate structures that span a larger space, then one could use a wireframe strategy. So in this example, what we've done is we've programmed the scaffold strand to wrap around this into this branched tree structure. And then we programmed staple strands to bridge these 2 terminal branches such that we end up with a double triangle. We actually did this in 3 separate test tubes with 3 completely different sets of staple strands. And then we programmed these terminal branches to link together to form this closed network, which maybe some of you might recognize as the Schlegel diagram for a wireframe icosahedron. Each one of these struts is programmed to be about 45 nanometers in diameter. So the entire structure is programmed to be about 100 nanometers in diameter, similar to the size of a clathrin-coated vesicle. Here's a macroscopic rendition of the folding process. So we fold the monomers and then heterotrimers them at a lower temperature and then look at them using our favorite method, transmission electron microscopy. And this is a 1 micron scale bar. So we can see, first of all, there's a bunch of particles that look like they could be about the right size. We see a bunch of aggregates, so self-assembly is not perfect, but that's self-assembly. Zooming in, we now have a 500 nanometer scale bar. We can see the characteristic hexagonal shape that might be reminiscent of icosahedral viruses. Zooming in some more, we have a 200 nanometer scale bar here. And we can really make out that this really does look like some kind of wireframe. And then now this is the highest effective magnification we can go to using negative stain. This is a 100 nanometer scale bar. It's the size we predict. We can really make out these triangular faces, these 5-fold vertices. Here's a particularly nice looking particle. In this orientation, we expect to see X, 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 X. And at least in my imagination, I see X, 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 X. So I think this is a testament to the power of DNA nanotechnology by simply exploring extrapolating the consequences of A pairs with T and C pairs with G, we can really generate fantastically complex structures. And in the future, we like to build things that are much more complex than this. Another method for building wireframe structures that span large distances was elucidated by the sculptor Ken Snelson. And he termed these objects floating compressions, in which you have rigid struts that are bearing compression that aren't touching each other, but they're connected by wires that bear tension. So he was inspired to do this by Buckminster Fuller before deciding that he hated his guts. But these objects have interesting properties. For example, they can, because the tension is globally distributed, one could apply a local pressure somewhere on the object, and that can lead to a deformation far away in the structure. And Don Ingber has been famous for exploring how cells use the principle of tensegrity in order to maintain their infrastructure and facilitate cellular communication. So Tim Liedl in the lab decided to implement this using DNA origami. And he chose to build, as his compressible struts, 13 helix structures. And for these tension-bearing wires, he used single-stranded DNA. Because as we know from physics, any kind of coil, any kind of linear polymer, if you make it long enough that it's floppy, it'll have an entropic tendency to shorten. Therefore, it's generating a force if you make it short enough. And here we can see the transmission electron micrographs of the objects. For most of them, you can't really see the single-stranded tethers. But you can make out that it does indeed have the desired three-dimensional, or appears to have roughly the desired three-dimensional structure. So everything that I've showed you so far involves using objects that are straight. And you can imagine, by analogy to macroscopic technology, that oftentimes it would be useful if we could build with curved elements Otherwise, the devices that we build might not be as efficient as we might like. So how can we build with curved objects? Well, I think this is going to be useful for our nanoscale objects as well. So one way to do that would simply be by taking the two ends of one of those sticks and pulling on them. And Tim Liedel Lab actually has done that using those floating compressions. By exerting enough force, you can get those struts to buckle. But I'm going to show you a related method developed by Hendrik Dietz. This is work that is coming out this week in Science. So this is our honeycomb lattice again, and I'd like to recall that the helices are cross-linked together along these planar faces by those holiday junctions. And if we want this structure to bend, there's a very simple-minded idea one might like to try, which is what if we design these helices just being shorter? So we scoop out material from the top, and we insert material on the bottom. Then what's going to happen? The top is going to contract, and the bottom is going to expand. So does this principle actually work, or do we just get misfolded DNA nanostructures? So here's the control. We have the curvature installed over a 98 base pair region. Again, this is an 18 helix bundle, and then we have default straight handles on either side just so we can see what's going on. We call this a protractor. This is the control, 0 degrees. We see these nice characteristic 3 stripes that we expect. Here he's installed, again, deletions on this face, insertions on this face, and we get a 30-degree angle. Here he has more deletions and insertions. We get a 60-degree angle. Here we have a 90-degree angle. Here we have a 120-degree angle. So I was already pretty impressed and amazed at this point, but Hendrik kept on going. Get a 150-degree angle. Here we have a 180-degree angle. This is quite amazing. This radius of curvature is close to what you see wrapping DNA around histones. In this diagram, we can see more closely the distribution of insertions and deletions. So as we insert more and more base pairs on the left and remove material from the right, we can see at 180 degrees, we're down to 6 base pairs per turn on the inside of this block. And we're up to 15 base pairs per turn on the outside. I was amazed that you could torture DNA this much and it'll still fold. I should say that when we get to those extreme angles, our folding yield starts to drop. So we're really pushing the limits of what DNA allows us to do. But I think it's still fair to say that DNA is an extremely malleable substance, and that we can use the enormous energy from DNA base pairing in order to get it to do amazing things. And the last thing I'd like to show you are examples of hierarchically connecting together these bent structures to create more complicated objects. So this is six hemicircles of increasing radius to make a spiral. These are both 6 helix bundle structures. This is the projection of a wireframe octahedron onto a circumscribing sphere. It has a diameter of about 50 nanometers. It looks suspiciously like the insignia of the institute that Hendrik applied for a faculty position for. And now he's the faculty member there. He's just started his lab in July. This is something we call the hyperbolic triangle. This is just recycled the 120-degree protractor that we programmed to homotrimerize. And then this is our favorite object. This is a symbol of technology, oftentimes used, especially exapted in the synthetic biology community. You'll see this on a lot of synthetic biology websites. But I don't think anybody's actually generated literally a structure like this, so we set out to do this. So what Hendrik did was to build a hemicircle with 3 square teeth that's programmed to homodimerize into a 6-tooth gear that has a diameter of 50 nanometers. And then he reprogrammed it to, instead of bending by 180 degrees, to only bend by 90 degrees. So now when you homotetramerize it, you get a 12-toothed wheel with a diameter of 100 nanometers. And so hopefully with these examples, I've persuaded you that we now have a general mechanism for building arbitrary 3-dimensional structures up to the size of about 2 ribosomes. And now we're pushing to build larger and larger structures. I'd love to tell you more about the kinds of applications we have in mind, and I thank you for your attention.